A strain of E. coli is cultured in a 15 meters cube mechanically adjusted bioreactor. Okay, so your volume of reactor is 15 meter cube. Under the operating conditions, the value of KLa is, so KLa is 0.17 second power minus 1. Oxygen solubility in the broth is 8 into 10 power minus 3 kg per meter cube. Then if the specific rate of oxygen uptake, okay, so QO2 is 12.5 millimoles gram power minus 1 half power minus 1, then what is the maximum possible cell concentration? So in this problem they are given all the data related to oxygen solubility. The cell concentration is calculated by relating oxygen uptake rate and oxygen transfer rate. Okay, so this oxygen uptake rate and oxygen transfer rate are equal when oxygen transfer is a rate limiting step in your process. Okay, when oxygen transfer is a rate limiting step in your process, then the rate of oxygen consumption, that is oxygen uptake rate, is equal to the rate of oxygen transfer. Okay, oxygen uptake rate is defined by QO2 into X. So QO2 is specific rate of oxygen consumption and X is your cell concentration. Okay, so oxygen uptake rate is equal to QO2 into X. So QO2 is specific rate of oxygen consumption by your cell. Then oxygen transfer rate. So let's know what is oxygen transfer rate. Usually your oxygen is introduced into a fermentation broth by sparging air through the broth. Okay, so oxygen transfer rate is the rate at which oxygen is transferred from the air, that is your gas phase, to the liquid, that is your medium. It tells us oxygen availability for our cells. So oxygen transfer rate is equal to KLa into C star minus Cl. So KLa is volumetric oxygen transfer coefficient and C star is saturated dissolved oxygen concentration, that is the amount of oxygen can be dissolved in the broth. The maximum amount of oxygen can be dissolved in the broth is your saturated dissolved oxygen concentration, and Cl is actual dissolved oxygen concentration in the broth. So in this problem, KLa, it is a given data 0.17 second power minus 1. QO2 is also a given data, that is 12.5 millimole gram power minus 1 half power minus 1, and C star minus Cl, the driving force, which is the difference between saturated oxygen concentration and your actual dissolved oxygen concentration. In this problem they have not given the value for C star minus Cl. Directly they have given oxygen solubility in the broth is equal to 8 into 10 power minus 3 kg per meter cube. Okay, that is your CO2. Oxygen transfer rate we can write as KLa into CO2. X is equal to KLa into CO2 divided by QO2. So in this equation, QO2 is given data, KLa is given data, then this oxygen solubility is 8 into 10 power minus 3 kg meter power minus 3. But we can't directly substitute all values in your formula because here KLa they have given in second power minus 1, but QO2 is in half power minus 1. We have to change the unit. Here also it is in kg, here it is millimoles. So we have to convert this weight into mole. Okay, KLa you have to change in hours, and this CO2 star you have to change in moles. Then meter power minus 3 also you have to change in liter because your answer is in the unit of gram liter power minus 1. Okay, so X is equal to, the formula is KLa into CO2 star divided by QO2. So KLa is 0.17 second power minus 1, it's a given data, and CO2 star is 8 into 10 power minus 3 kg per meter cube. Then QO2 is 12.5 millimole gram power minus 1 half power minus 1. Okay, first change this 0.17 second power minus 1 into half power minus 1. Okay, so 1 hour is equal to 3600 seconds. So 1 second is equal to 1 by 3600 hour. So we need in second power minus 1. So inverse everything. So 1 second power minus 1 is equal to, if you inverse this, you will get 3600. So 1 second power minus 1 is equal to 3600 and half power minus 1. So 0.17 second power minus 1 is equal to 0.17 into 3600. Okay, so 0.17 second power minus 1 you can write as 0.17 3600 hour power minus 1. Next we have to convert 8 into 10 power minus 3 kg meter power minus 3 to millimole liter power minus 1. To calculate mole, you should convert kg in gram, because mole is equal to weight in gram by molecular weight. Okay, so for that convert kg in gram. So 8 into 10 power minus 3 into 10 power 3. 1 kg is equal to 1000 gram. Okay, so weight by molecular weight. So your molecular weight of oxygen is 32. Okay, then you have to change in millimole. Okay, so 1 mole is equal to 1000 millimole. So once again you have to multiply with 10 power 3. Okay, so this 10 power 3 is kg to gram. 1 kg equal to 1000 gram, and this 10 power 3 is mole to millimole. That is 1 mole is equal to 1000 millimole. Okay, you are dividing with 32 because you are converting gram to mole. So weight divided by molecular weight of oxygen. Okay, then meter power minus 3 should be converted to liter. Okay, as we know that 1 meter cube is equal to 1000 liter, that is 10 power 3 liter. So 1 meter power minus 3 equal to 10 power minus 3 liter power minus 1. Unit is changed from kg to millimole and meter power minus 3 to liter power minus 1. Cancel 10 power minus 3, 10 power plus 3, 10 power plus 3, 10 power minus 3. So you will get 0.17 3600 into 8 divided by 32 into 12.5. Okay, 32 into 12.5. Okay, so if you simplify this into 12.5, you will get 12.24. Okay, so the answer is 12.24. So here millimole cancel it out, har cancel it out. So liter power minus 1, gram power minus 1. So you can write this as x is equal to 12.4 gram per liter. Okay, thank you for watching my videos. If you like my videos, please do like, share and subscribe. Thank you.